are the knife group. My name is Megan Howler.
And my name is Maliha Asher.
My name is Nick Kinsella.
And my name is Jade Bell.
So during this semester, we discovered 3 new phages called Reckman, Nosy Nova, and Decay Blade. Reckman was found in Joe Farmer Recreational Center in Allen, which is where it got its nickname Rec. Nosy Nova was found at the Frisco Collin College campus under a red tree and is a namesake of Jade's cat. Decay Blade was discovered in Nick's backyard in Dallas and named after the decaying tree stump and one of Nick's favorite movies. All 3 of these are cyphroviruses, which is the largest viral family on Earth and includes members infecting both bacteria and archaea.
So phages are a virus that are able to infect bacteria cells in order to replicate and survive. Another name for phages are bacteriophages, which means bacteria eater, and this is because they are the natural enemies of bacteria. Phages are found in many different bacterial colonies, but mostly in soil or seawater. Ours were found in soil. Phages also exhibit 2 different life cycles, the lytic cycle, the lysogenic cycle. In the lytic life cycle, the phages are going to inject their DNA into the cytoplasm of the bacteria host cell so that they can then replicate inside of the host. Whenever they are finished replicating, they will then lyse the bacteria cell, thus breaking the cell open in order to spread the new phage material. And the other one, the lysogenic life cycle, the phage will inject their DNA directly into the bacteria DNA, where the then will replicate and then divulge into the lytic life cycle, where it will lyse the cell and spread that new phage material. Phages are also specific to certain bacteria. For example, the phages discussed here are specific to Microbacteria foliarum.
So we collect our samples and then we isolated them and then did serial dilation to find out more, individual phages and then follow more protocols to more results.
So we have included our pictures of our phages here with our scanning electron microscope image of the phage, our spot titers, and then the digest of our electrophoresis. These phages were found, phages Reckman and Nosy Nova were found via direct isolation, while phage Decay Blade was found via enriched. Here we have our spot titers, and if you see that these plaques are more turbid and kind of boggy, as well as these, meaning that those are part of the lysogenic life cycle, while phage Reckman has more of clear plaques, meaning that this phage is of the lytic cycle. My plaques, I had plaques ranging from 1 to 2 millimeters in diameter, and I had a titer about 1.6 times 10 to the 9th.
So on my plaques, mine were uniform plaques at 3 millimeters in diameter, and my titer went to 1 times 10 to the 11th.
And then my plaque, it was ranging from 1 to 4 millimeters, displaying a titer of 3.3 times 10 to the 9th.
So here with our gel electrophoresis results, the DNA of each phage was digested and prepared to run on the gel. The DNA was cut with 3 different enzymes, the Hy3, NSP1, and SAC2. Along with that, we also included a ladder so we can kind of see what the sizes of the different DNA fragments of the phage were. We have Hy3 and SAC2 will cut DNA between the various combinations of G's and C's, while the NSP1 will cut equally more along all 4 of the nucleotides. So like here with phage Nosy Nova, you can see that NSP1 cut more versus the SAC2, and I have none on the Hy3. And then with phage Reckman, we can again see more cuts along the NSP1 and the SAC2 versus the Hy3. And then ending with phage Decay Blade, we actually have more cuts along the SAC2 phage or enzyme versus the Hy3 and the NSP1.
So with the discussion of phage therapy is actually at an all-time high right now. as scientists are steadily finding more ways that phage can be beneficial to us. Phage therapy involves the use of natural intact phages, also known as bacteria viruses, for treatment of bacterial infections. While phage therapy is not new, its use in people and animals has not been fully researched, as it is still not FDA approved. The reason that phage therapy is gaining so many popularity is because phages are more specific to their bacterial targets than antibiotics are, which causes less harm to any of the beneficial bacteria strains. Phages, although they're not considered living, they are very dynamic and always evolving, thus they are not easily manipulated and can act as more of a disruptor to the body. Phage is the only known parasite, though, to target specific bacteria, and in theory, it will spread if there are cells to infect. Then when the pathogens are eliminated, they cannot survive without a host, later to be disregarded. Phage work can be very precise, so it is important to follow the given protocols to avoid any error. All protocols followed were part of the Phage Discovery Guide. Throughout the semester, our group had to redo a couple of the protocols, which might have caused inaccuracy in our results. I guess to generate well-isolated plaques, we used protocol 6.1, which I ran into some trouble with. It was important to mix the plaques with M. coli, which I did not do, which caused no bacteria to grow, so I had to redo it 3 times.
And for the digest and gel protocol, which was protocol 10.1, I accidentally skipped over page 79, which was phage prep. So that included incubating at 65 degrees Celsius for 10 minutes and then quickly moving it over to ice. I guess I just didn't read the page right. I thought it started on the next page, so I started off by centrifuging, which I think could have affected, the bacteria's growth.
And then for mine, I had trouble finding any phage activity with my first 2 samples that I had isolated from. And so because I couldn't find any there, I had to go to my 3rd sample, which is where I was able to find phage matter. And then here in my scanning electron microscope image, it does seem appear to be a lot cloudier than decay blade and Reckman, and I believe that's because I didn't wash my sample thoroughly enough. So I think there's still some salt components left on it.
And these are our references right here. And yeah.
Thank you.
Thank you.